Hi everyone, welcome to, this will be week 6 now, or week 7, okay. I changed the order around a little bit, so not that it really matters. My computer, I just got fixed, it had the other presentation on. So, we are looking at rates of reaction, catalysts and enzymes, and it is a lot to get through. The entire year 11 curriculum in 10 sessions, but we're going to do our best. Okay, rate of reaction. Anyone tell me what this means? Why is that video showing backwards? So anyone know what rate of reaction? I do not want to mirror my video. Go, it's better. It's how quickly a reaction proceeds. So change in the amount of products over time. It could also be reactant, so reactants or products over time per unit. So the reaction, it is fastest at the start. So this is when the products, sorry, the reactants are at their highest. As they're used up, the reaction goes down. The rate of reaction goes down. So the amount of product depends on the amount of reactants used. So if we were to use a graph, and this being the product, the amount of product over time, it would look something like this. So who can tell me why it would taper off like that? The idea is why the reaction would just basically stop. So the reaction tapers off like that because we run out of reactants. So we run out of reactants, right? That's why the reaction stops. So this is a bit of simple, typical chemistry using simple language. But we have fast and slow reactions. Anyone think of examples of a fast or slow reaction? Fast is combustion, so burning things. Slow would be rust. So slow happens over, Gizzy, me too, something morning. Fast is combustion, slow is rusting, fermentation. So things that take a decent amount of time. Ripening of tomatoes. We use a gas now. We use a gas to ripen tomatoes, and it happens virtually instantly. So fast reactions. So combustion, burning of petrol. And other fast reactions will be if you dropped a piece of sodium, I'd Google it or YouTube it, whatever you want to call it. If you drop a piece of sodium into water, you get a nice big explosion. So that's quite a fast reaction. Slow reaction, photosynthesis, rusting, fermentation. So things that we can observe, and again, this is definitely in the curriculum, things that we can observe to measure the rate of reaction are precipitates. So sometimes we have to measure rates of reaction by eye. It's not possible to do it the other way. So change in mass of either reactants and products. Color change. So one of the other examples for a fast reaction is actually the fading of a t-shirt in the sun. So temperature changes, either exothermic or endothermic. We have pressure changes. And the biggest one is the volume of gas produced. So that's the one we use to calculate the rate of reaction of a lot of reactions. So the way that we actually calculate it, I'm going to keep it fairly simple. So we do have 3 decent sized topics. So the rate of reaction, it's a fairly simple equation, but actually determining this is quite difficult. Just going to call it the rate equals the quantity change of reactants or products over time. Sometimes we have measurable quantities. Other times we're talking about quantities that are too small. So we can make some assumptions when we're looking at a reaction. So we can say that in a chemical reaction, 2 things happen. Quantities of reactants reduce and the quantity of the product increases. Fairly simple and straightforward. We know those 2 things will happen. The law of conservation of mass and energy tells us that will happen. So we could then theoretically measure the rate of reaction by measuring the decrease in the reactants over time or measuring the increase in the products over time. this is where it gets tricky. Sometimes we can measure things, other times we can't. The easiest is to measure the mass. So you're trying to react two solids, a solid and a liquid. You just measure the mass of the object on a scale whilst the reaction's happening, and you plot it getting smaller, and you divide that by the time, and then you would convert it into moles. So we can use the mass, we can use the concentration. So if you're using metals or salts, you can actually measure the conductivity, which is an indicator of the concentration of a salt or metal in a solution. Or the other one is the volume of gas produced. This is the one they normally use, because that's quite easy to capture. It's a quite easily measurable quantity. There's a number of things that affect the rate of reaction. Any ideas, Mark? Anybody? Yeah. Okay. So the biggest one, if you're using a solid reactant, is the size of solid. And that's because the bigger, if you have one big block of sodium chloride that we're trying to react with something, it would react slower than if you ground that up into a fine powder. So concentration of the reactants, up to a certain point, the more reactants you have, the faster the reaction will progress. The temperature, higher temperature equals higher rate of reaction. In grade 12, you will learn about why that happens. Pressure, believe it or not, it's not a one size fits all. Depending on the chemical reaction you're dealing with, sometimes you want high pressure, sometimes you want low pressure. And funnily enough, catalysts. So what is a catalyst? Any ideas? Let me have a think about it for 1.5 minutes. What's a catalyst? No, it's either nothing to do with eyes. Well, you're thinking about cataracts. All right. Right, I need to get rid of rate of reaction. Now we have catalysts. Catalysts increase the rate of reaction. That's a lie. They can also decrease the rate of reaction if you want to slow things down. But for the purpose of this unit, we are just looking at catalysts that increase the rate of reaction. So when we talk about a chemical reaction, every chemical reaction activation. Every chemical reaction has an activation energy, or the amount of energy required for a chemical reaction to take place. So the catalyst reduces the activation energy. And the technical way to describe it is it provides an alternate reaction pathway. So if we were to do a reaction, rate of reaction over time, so something like that. So this is the energy required. We then have our catalyst. So the amount of energy required is significantly reduced. There's a few important things you need to know about this. Is the catalyst does not take part in the reaction. So it is either, it's not consumed, it's still there in the same form it was at the end. Catalyst does not take part in the reaction. Again, this is kind of segueing into our next. These catalysts are highly specific. So now think about this in terms of a living system. So living systems are supposed to be as efficient as possible. But many of the reactions that take place in the human body, a little bit of biochem here, normally would happen too slowly in order for us to survive. Take 30 seconds to think about I'll take a minute. What do we have in our body that allows us to survive by lowering the activation energy? I'm going to do So we're talking about enzymes. Enzymes are biological catalysts. Wish I had some music to show you how it works, but this stuff here, peroxide, H2O2, O2 minus, so on and so forth, is where this diet fad of eating antioxidants came from. The body already has ways of dealing with it, but it's too slow. This stuff is super toxic inside the body. It's produced in our cells normally. And what we need is an enzyme to break down the hydrogen protein. So we use an enzyme called, well, all living things use an enzyme called catalase to break down H2O2. If we didn't have this enzyme, H2O2 would build up in the body and we'd be dead very quickly. Yeast also have it. In year 11, you should do an experiment where you take beef liver. Go away. Sorry, good child's screaming into the window. You'll take beef liver, which is where catalase is produced. You'll mix it with hydrogen peroxide. And you'll watch the gas bubbles produced, and you'll see the enzyme at work. Now, biological catalysts, they are extremely specific. They are made up of proteins. Sometimes they'll have metal attached, metalloenzymes. Sometimes they'll have salts. But they are super specific. So, what does this mean? If we didn't have the enzymes, none of the chemical reactions that happen in our body would happen fast enough for us to survive. So just like the catalysts, these are just biological catalysts. They increase the rate of reaction by reducing the activation energy. These are biological molecules, and they do not last forever. Whereas a catalyst will always be there. Sometimes an enzyme can be broken down. Okay, so now I'm going to draw a ridiculous picture. Everyone's going to laugh. Okay, so this is our enzyme. Looks like a kidney, I know. So this is our enzyme. And the most important part about the enzyme is the active site. So we have our hydrogen peroxide, which fits perfectly into that active site. Upon binding to the active site, the hydrogen peroxide is broken up into H2O and O-. So now it's perfectly safe and harmless. So the enzyme has an active site matches perfectly, this is catalase, matches perfectly to the peroxide, hydrogen peroxide, H2O2. Oxygen-free radicals, whatever you want to call it. There's 7 different classes, and I'm not going to write them all down, I'm just going to skip them, otherwise it'll be further. And their names are based on what they do, and you don't need to have a massive understanding. So oxidoreductase, transferases, hydrolases, isomerases, ligases, or translocases. And you don't need to know them all, but the names are indicative of what they do. So we have two hypotheses, lock and key, and the induced fit. So take 30 seconds to have a think about what those two things could mean. Yeah. Okay. So lock and key, let's have a master key, assumed one enzyme for one product. I just have had the most massive mind blank. Oh, that's the technical name. That it's locked, one enzyme does one job. The active site does not change. The induced fit model says that one enzyme does multiple jobs. I'm trying not to get too biology here. One enzyme, one substrate. There we go. Just going back to my mark. One enzyme, one substrate. So the induced fit, one enzyme does multiple jobs, or the active site changes, alters its size to do multiple jobs. Enzymes are probably one of the most, I guess all the biological bits are, non-biological things are important. But without these enzymes functioning correctly, we end up with genetic diseases. The jury is still out on which one of these completely lost train of thought. Sorry. So we have so let's get a little bit biology and let's look at these genetic diseases because it is important in the context of chemistry to understand. This active site, again, you'll learn this in grade 12, it has to be that specific shape or a very similar shape, even in the induced fit model. When you have a genetic abnormality, it may change the protein to have that shape of an active site. Now, if you're unlucky enough for that to be in one of your genes that encodes for an enzyme that creates mucus, you would get cystic fibrosis. Because the enzyme, it might work a little bit, but it doesn't work properly. So let's finish looking at the activation energy. So we know that if we didn't have the enzymes, the chemical reactions needed to keep us alive would happen so slowly that life wouldn't be possible. And all the enzyme does, when it binds, it changes the shape. Changes the shape of the structure, which allows the reaction to occur at a faster rate, or, as we know, it lowers the activation energy. Enzymes are not used. Not used in the reaction. They do not interfere with the energy change of the reaction. So it's an interesting crossover here between bio and chem. Funnily enough, it's called bio. Some enzymes require cofactors. So examples of this: zinc, iron, vitamin A, B12, so on and so forth. So all those healthy things and all those multivitamins, they all allow enzymes to go about their reaction. So these are called coenzymes or cofactors. So biotin, which I think is B3, B12, folic acid, B2, B3, B4 are all what we call cofactors for enzymatic reactions. So they say if you have vitamin B12 deficiency, you have low energy, tired all the time, because vitamin B12 is a cofactor in the production of energy. If you don't have it, the enzyme that catalyzes the reaction and without it, we don't get any energy produced. And we'll finish up on this. So, although this is the problem with enzymes, there are so many factors that will affect the rate of reaction. So when you get sick, you get a virus, your body spikes your internal temperature, quite literally to try and bake the virus out. So the virus's enzymes can't work at higher than 39 degrees. So temperature, so affected by the pH. So they are designed to work pH 6.8 to 7.2. Any higher or lower, they start to not work properly. Temperature, pH, concentration of enzyme, concentration of substrate, and these are dependent on the concentration of the other. So if you just ramp up the concentration of the enzyme, it's gonna the rate of reaction will taper off. Same thing if you increase the concentration of the substrate. They have to be increased together. Temperature and pH will change the shape of the active site. The concentration of the enzyme and the concentration of the substrate, and concentration of the complex, which is the enzyme bound to the substrate. So temperature, pH change the shape of the active site. Concentration of these three will effectively crowd the enzyme. So there'll be too much and none of the unbound enzyme or substrate will be able to get close enough to each other. There's inhibitors. So a lot of things like organophosphate fertilizers. Alcohol's main metabolite, acetaldehyde, is an inhibitor to breaking down alcohol. So the more alcohol a person drinks, the longer it actually takes the alcohol to be metabolized. So these are inhibitors. A lot of poisons that are used to poison people, I guess, are actually enzyme inhibitors. So they competitively or non-competitively bond to the active site of the enzyme and block it from reacting with the substrate. All right, I think we'll leave that there for today. That was a lot to go through.